Welcome to the Fundamentals of Pathology. The first section that we want to cover is growth adaptations, and this is obviously the first section in Chapter 1 of your text. A few basic principles before we begin. The first is that an organ is in homeostasis with the physiologic stress placed on it. So we can take any example. Take the heart, for example. The heart has the function of pumping against the systemic blood pressure, and it pretty much is in homeostasis when it deals with that physiologic stress. Now, if there is an increase in stress, or a decrease in stress, or a change in stress on an organ, then the organ will undergo growth adaptations. And these growth adaptations is what we want to discuss over the next few minutes. And generally speaking, the growth adaptations are very high yield for examinations. Now, the first of the growth adaptations is hyperplasia and hypertrophy. And the idea here is that if there's an increase in stress on an organ, this will then lead to an increase in organ size. Basically, if the organ has to do more work, the organ will become bigger in order to do that excess work. Now, how will the organ become bigger? Well, there's two ways. One way by which the organ could become bigger is to increase the size of the cells of that organ. And an increase in cell size is actually called hypertrophy. So one way by which the organ becomes bigger is to increase the cell size, i.e. hypertrophy. Another way by which the organ can become bigger would be to increase the number of cells of the organ, and an increase in number of cells is called hyperplasia. So basically speaking, these two mechanisms, hyperplasia and hypertrophy, allow for the organ to increase in size in the face of an increase in stress. Now, it's not enough to simply know the definition of hypertrophy and hyperplasia, but we've got to go one step further, and examiners like to ask about about the mechanism that underlies these changes. So let's take hypertrophy first. Recall that hypertrophy is what? Hypertrophy is when we take a cell and we make that cell bigger. We increase the cell size. Now, if we're going to increase the cell size, a couple important things. First of all, recall that a cell has a skeleton, which is called the cytoskeleton. And that cytoskeleton is what gives the cell its size and its shape. If we want the cell to become bigger, we're going to have to increase the production of that cytoskeleton. And the cytoskeleton is predominantly made of protein. So one of the very important mechanisms for hypertrophy would be protein synthesis. Now, obviously, in order to produce proteins, genes are going to need to be activated. So you could put this story together by basically saying that one mechanism of hypertrophy is gene activation and protein synthesis. Another important thing that will have to occur in hypertrophy is the production of organelles. If the cell is going to go from this size to this size, then it's going to need the organelles that are necessary for cellular function. For example, mitochondria is important for cellular function, and so you will need to increase the number of mitochondria in a cell if we increase its size. So production of organelles is also an important mechanism of hypertrophy, and both of these go hand in hand. So that's why I say here in your text that hypertrophy involves gene activation, protein synthesis, and production of organelles. Now, hyperplasia, on the other hand, recall hyperplasia being an increase in cell number. So an increase in cell number is going to involve the production of new cells. And new cells will be produced from stem cells. And that's, I think, pretty straightforward to understand. Generally speaking, hyperplasia, which is an increase in the number of cells, and hypertrophy, which is an increase in the size of the cells, usually occur together. So let's take a simple example. When a woman becomes pregnant, the uterus is going to increase in size. Obviously, the uterus has to increase in size because it needs to provide space and a home for the developing fetus. Now, that uterus is going to increase in size in two ways. First, the smooth muscle of the uterus will undergo hyperplasia, i.e. there will be increased cells produced. And second, the smooth muscle of the uterus will undergo hypertrophy, so the cells will also become bigger. And generally speaking, hyperplasia and hypertrophy will actually occur together. One important exception to this general principle is permanent tissues. Now, permanent tissues, they do not have stem cells, and because they do not have stem cells, they cannot make new cells. Hence, because they can't make new cells, they won't be able to undergo hyperplasia. And so they'll undergo hypertrophy only. This is a very high yield topic. So let's take a step back and remind ourselves that in the human body there's basically 3 permanent tissues. The first of the permanent tissues are cardiac myocytes. The second permanent tissue is skeletal muscle. And the third permanent tissue is nerve. These 3 tissues, cardiac muscle, skeletal muscle, and nerve, cannot make new cells and therefore will undergo hypertrophy only and cannot undergo hyperplasia. Here's a classic picture that likes to show up on exams. This is actually a picture of both the left ventricle and the right ventricle. Here we can clearly see that the left ventricle is very thick and that the right ventricle is basically exhibiting a normal thickness. Now, I could show you this picture and then I could ask you, what's the mechanism by which this change has occurred within this organ? And the answer would clearly be that this change, this increased thickness of the wall of the cardiac muscle has actually occurred via hypertrophy because the heart cannot undergo hyperplasia. Another important principle regarding hyperplasia is that pathologic hyperplasia can progress to dysplasia and cancer. Now, what do I mean by pathologic hyperplasia? Pathologic hyperplasia is when hyperplasia occurs due to some underlying pathologic process. And this pathologic hyperplasia can progress to dysplasia and cancer. Maybe we should take a step back and contrast pathologic hyperplasia to physiologic hyperplasia. An example of physiologic hyperplasia would be pregnancy, where due to some physiology of the organism, hyperplasia occurs. And there is no increased risk for cancer with physiologic hyperplasia. However, with pathologic hyperplasia, there is an increased risk for dysplasia and cancer. Classic example, endometrial hyperplasia. You're well aware from your pathology lectures that the endometrium, or from your physiology lectures, that the endometrium grows with exposure to estrogen, and it sheds, and it develops and sheds with exposure to progesterone. Now, if the endometrium is overexposed to estrogen for long periods of time, the endometrium will actually undergo endometrial hyperplasia. And this endometrial hyperplasia can eventually progress to the formation of endometrial carcinoma. And we'll talk much more about that when we do the gynae lectures later in this series. Another important point to be brought up here is that there is one exception, or one very classic exception to this general rule, and that is benign prostatic hyperplasia, or BPH. Now, benign prostatic hyperplasia is a pathologic hyperplasia. However, it is not related to cancer, and there is no increased risk of cancer with BPH. So this again makes for a nice question, examiner's question, because of the fact that it's an exception to a general principle. Okay, the next growth adaptation is atrophy. Now, the idea with atrophy is that there is a decrease in stress on the organ. So if there's less stress on an organ, the organ will actually decrease in size. And that decrease in size is called atrophy. Now again, really no rocket science. If an organ is going to decrease in size, there's really only 2 ways for that to occur. Number one would be for the size of the cells that make up the organ to decrease, or number 2 would be for the number of cells within that organ to decrease. If the size of the cells decreases, the organ would decrease in size, and if the number of cells decreases, the organ would also decrease in size. Both of those happen to be called atrophy because they usually both occur together. One, again, it's not sufficient to simply know the definition of atrophy. The examiners like to go after the mechanism of atrophy, and so let's touch on that briefly. Well, remember that atrophy is going to occur in 2 ways. The first would be to decrease the number of cells. And a decrease in number of cells is going to occur via apoptosis. Apoptosis is a mechanism of cellular death that we'll discuss in more detail later on in this chapter, but it's very important to know that a decrease in cell number will actually occur via apoptosis. A decrease in cell size is going to occur by 2 primary mechanisms. The first is something called the ubiquitin proteasome degradation pathway. Now, what's the idea here? Well, recall that if you've got a cell that's this big, and you want the cell to shrink down to something that's smaller, in order for the cell to shrink, this skeleton, called the cytoskeleton, that was maintaining the size and shape of the cell, has to be broken down so that the cell can become smaller. The mechanism by which the cytoskeleton is broken down is called the ubiquitin proteasome degradation pathway. Ubiquitin is a protein, I kind of think of it as a post-it note. It's a protein that gets posted onto the intermediate filaments of the cytoskeleton, and the proteasome is a organelle present within the cell that recognizes ubiquitin tagged proteins and then trashes those proteins and destroys them. And so the ubiquitin proteasome degradation pathway is a mechanism for destroying the cytoskeleton, and it's a high yield point to be aware of. Now, at the same time, in order for the cell to shrink in size from this size to, for example, this size, you're going to end up requiring less organelles within the cell. And so some of the cellular components are going to have to be consumed, and the mechanism by which cellular components are consumed is called autophagy. And basically what happens here is that the cell consumes its own components in vacuoles. So for example, if this is the cell here, little vacuoles are created, and those vacuoles, they fuse with lysosomes, and the lysosomes contain the hydrolytic enzymes that are necessary to break down and destroy products into their simple building blocks. And so basically what will happen is that a autophagic vacuole will be formed, and the cellular components will be consumed and then destroyed by the lysosomes. So two mechanisms by which the cell size decreases. Number one, ubiquitin proteasome degradation pathway, and number two, autophagy of cellular components. The third growth adaptation, which is also relatively high yield, is metaplasia. Now, the idea here